Okay, welcome. This week we're going to be talking about the tissue level of organization. So what is a tissue? A tissue is an accumulation of specialized cells that are coming together to perform a specific function. Histology would then be the study of tissues. So remember when we talked about the levels of organization week 1, we started off with the chemical level of organization, worked our way up to the cellular level. Now we're at the tissue level, and then we'll go to the organ and organ system level, which makes up the organism. The 4 different types of tissues are epithelial tissues, connective tissues, muscle tissues, and nervous tissues. This week we'll be talking mostly about epithelial tissues. Next week we'll dive into connective, muscle, and nervous. Epithelial tissues can be separated into epithelia and glands. Epithelia are going to cover our external surfaces and line all of our internal surfaces. Glands will produce fluid secretion, so we'll talk about each of those. Some of the functions of epithelial tissues: they provide physical protection, so they protect us from abrasion, dehydration, and destruction via chemicals, bacteria, viruses, anything we come in contact with in the world. Our skin is going to protect us from, and our internal body surfaces will protect us from. Epithelial tissues also control permeability, so they act as a barrier that can be regulated or modified for the tissue's needs. This will change based on hormones, physical stressors, etc. So one good example is calluses. Calluses will form on your skin in areas where there's a lot of traffic, and when calluses form in those places, they will cause a more difficult barrier for things to get through. Epithelial tissues also provide sensation, so they have a large sensory nerve supply. There's also neuroepithelia that are specific to providing us with certain senses, such as smell, taste, hearing, etc. We'll talk about those when we get to the senses. And then specialized secretions. Those are going to be our glands. The gland cells will produce secretions, and they will be brought to either the surface of the epithelia or to the bloodstream to be circulated throughout the body. Epithelial tissues have certain characteristics as well. They have polarity because they have 2 different surfaces: the apical surface that's going to be in contact with the external or internal environment, depending on where these epithelia are. If it's the skin, then it would be the external environment. And if it's on the inside of our bodies, it would be, our internal environment. They also have a basal surface that's going to attach to other tissues like connective tissues. And because there's such a difference in function between these apical and basal surfaces, that's what allows for this polarity of the tissue. The tissue also has cellularity because these cells are bound so closely together in comparison to other tissues through cellular adhesions, which contributes greatly to their function. They also have attachment, so these epithelial cells are all bound to a basement membrane. The basement membrane will resist stretching and tearing of the epithelia. Epithelial tissue is avascular, so it has no type of vascularization, no arteries, no veins, no capillaries, so it receives its nutrients via diffusion. It also has regenerative capabilities, so epithelial tissue is continuously replaced more than other tissues because it's constantly in contact with surfaces where they can be abraded, chemicals that can damage it, and things of that nature. So certain epithelial cells can be specialized to move fluids for protection and lubrication, move fluids to control permeability, so some have microvilli to allow for more absorption in places where absorption is more necessary, such as the small intestine. Some have cilia, and at that point they're called a ciliated epithelium, to help move debris, such as in the respiratory system. And some are specialized to produce secretions that provide physical protection or act as chemical messengers, such as the glands. So how are these cells connected to each other? By cell adhesion molecules, or CAMs. CAMs are going to bind to each other and extracellular materials to allow for these cells to be pretty close together. Then there's also 3 different types of cell junctions: gap junctions, tight junctions, and desmosomes. Gap junctions are connections between 2 cells by these proteins called connexons. These are essential for contractions in muscle, so they're found in other tissues other than just these epithelial tissues. They're found in muscle tissues as well. Tight junctions are also known as occluding junctions. They're held together tightly by adhesion belts. They are going to prevent the passage of water and solutes, so they're very important in gastrointestinal tissues when you need all of these nutrients to be absorbed from digestion. If these tight junctions are not functioning properly, that's what leads to things like irritable bowel syndrome and what is generally known as leaky gut syndrome. Desmosomes are another type of cellular connection, strong connections that resist stretching and twisting. They're very abundant in the superficial layers of the skin because they're more tightly bound together than these other junctions are. So it allows for the skin to be waterproof and to not allow quite as many substances in and out. Spot desmosomes are these small discs that are connected to intermediate filaments between cells. And hemidesmosomes are similar. They're about half the size of the spot desmosome, and they attach the cell to the extracellular filaments of another cell. So all of these epithelial cells are attached to the basement membrane. If they're not attached to the basement membrane, they can die because they're not vascular, so they would not be receiving any nutrients if they weren't attached to the basement membrane where nutrients could diffuse through. There are 2 different parts of the basement membrane, the basal lamina and the reticular lamina. The basal lamina is actually superficial to the reticular lamina, and it acts as a selective filter. The reticular lamina gives the basement membrane its strength. So because these epithelial tissues are constantly exposed to harsh environmental conditions, it needs to constantly replace itself. So stem cells are necessary to keep dividing and forming these daughter cells that can provide for more epithelial cells. So we're going to talk about different types of epithelia. They can be sorted based on shape of the cells and the number of the layer of cells. So the different shapes are squamous, which is more of a flat type of cell, cuboidal, which as it sounds is more cube-shaped, and columnar, which is more rectangular. As far as layers are concerned, simple means that there's one layer of these cells, and stratified means that there's more than one layer. So we'll start off with squamous epithelia. So they're thin, flat, irregular. The simple squamous is the most delicate epithelium, so it provides for a lot of gas exchange. So these are generally found in the respiratory system and the blood vessels. It also can be found in the linings of the body cavity and the heart. Mesothelium is a type of simple squamous epithelium, and it lines the body cavities, and then the endothelium is going to line the surface of the heart and the blood vessels. The stratified squamous epithelium is important in areas where there's a lot of mechanical stress, so places like the skin, the mouth, the esophagus, vagina, and anus. There are 2 different types of stratified squamous epithelium. You have keratinized and non-keratinized. Keratinized means that there's a lot of keratin provided in the epithelia, so it makes it more waterproof. So you'll find this in areas that are supposed to be kept dry, like the skin. Non-keratinized epithelium means that there's not a lot of keratin, so these are in areas where we don't want waterproofing. We actually want these tissues to be rather wet, and they have to be kept this way or they can deteriorate. So examples of that are the oral cavity, the pharynx, the esophagus, the anus, and the vagina. With cuboidal epithelium, the simple cuboidal epithelium is specialized for secretion and absorption, so we'll find this a lot in glands and kidney tubules. And the stratified cuboidal is rather rare. It can be found in sweat ducts and larger mammary ducts. Transitional epithelium is a stratified epithelium that changes between squamous and cuboidal in shape. So this will depend on how much it's being stretched. It tolerates stretching without damage. We'll find transitional epithelium primarily in the bladder. So these cells will be able to tolerate stretching of the bladder when it's more filled with urine to be excreted without damaging the bladder itself. With columnar epithelium, we have 3 types. So simple is, again, specialized for absorption and secretion. We'll find this a lot in the gastrointestinal tract, like in the small intestine, stomach, and the large intestine. Pseudostratified is called that because it looks like it has multiple layers, but in fact it has one, and the cells are going to look like they're different shapes. All of the cells will contact the basement membrane just like they do in simple columnar. Typically, pseudostratified columnar epithelium is ciliated, so we would then call it pseudostratified ciliated columnar epithelium. And it can be found in the nasal cavity, the trachea, bronchi, and the male reproductive tract. Stratified columnar epithelium is also rather rare, just kind of like the stratified cuboidal, and this can be found in the pharynx, epiglottis, anus, urethra, and several excretory ducts. Glandular epithelia are specialized for secretion, so we have endocrine glands versus exocrine glands, and sometimes they're even both. Endocrine glands are going to secrete their substance directly into the bloodstream, whereas exocrine glands will secrete it into a duct, which will bring it to whatever tissue that is the target tissue. So endocrine secretions are known as hormones, and they're considered to be ductless glands because their products are secreted right into the bloodstream rather than into ducts. Some examples are thyroid glands, thymus, pituitary gland. There's so many. The pancreas is also an endocrine gland, but it is also considered an exocrine gland. Exocrine glands are going to produce exocrine secretions via ducts. Some examples are digestive juices and enzymes, sweat, tears, and breast milk. Glands can be unicellular or multicellular. Unicellular glands primarily have goblet cells, which produce mucus through a protein called mucin, and multicellular glands are going to produce larger products like saliva from salivary glands. Multicellular glands tend to have a variety of different structures. So the structure of the duct can either be single or compound. That should be simple or compound. If it's simple, then it's a single duct and it does not divide. And if it's compound, then the duct divides one or more times. The secretory portion of the gland can be alveolar or acinar or tubular. Sometimes it can even be a combination of the two. So tubular is when the glandular cells form tubes. Alveolar or acinar is when they form blind pockets. In the relationship between the duct and the glandular areas, it may be branched, and if it's branched, then several secretory areas are going to share the same duct. So here is a picture of some of those different types of glands. The glands can also have different methods of secretion. We're going to go through each of these. Merocrine secretion is when the product is released via exocytosis. It's the most common secretion method. It's usually found in salivary glands or merocrine sweat glands, and those sweat glands are the type that when you're exercising and you're sweating, that's merocrine sweat. It typically doesn't have an odor. It's just more of a liquid perspiration. It wouldn't cause stains in your clothes. In merocrine secretion, the cell is not damaged when the product is secreted. In apocrine secretion, it's going to involve the loss of the cytoplasm in the secretion. So the cell itself isn't breaking down completely, but the cytoplasm is lost. So an example of apocrine secretion is milk production, which is actually a combination of merocrine and apocrine sweat production. This is more of your puberty-related sweat. This is the sweat that would stain your clothes yellow and have more of a body odor to it. And in holocrine secretion, your gland cell is going to become destroyed with the secretion. So the entire cell is destroyed. It becomes packed with all these secretory vesicles and it bursts, killing the cell. An example is sebaceous glands, that it's going to produce this sebum or oil for your skin or for your hair. And then the types of secretions themselves can be either serous, mucus, or mixed. If they're serous, then the solution is more watery and it contains enzymes. An example would be the parotid gland, which is one of our salivary glands. It's our largest salivary gland. Mucus is going to produce mucus. So an example of that is the sublingual and submucosal glands of the small intestine. The sublingual gland is another one of our salivary glands, so it'll have a more mucus-like secretion. And then lastly, they can be mixed. So when they are mixed, they can be both serous and mucus. And an example of that is the submandibular gland, which is our last type of salivary gland. So for our salivary glands, we have one that's serous, one that's mucus, and one that's mixed. So we'll pick up with connective tissues next week, and we will also start the integumentary system.